Hello everyone, welcome to today's live broadcast, the 100,000 Genomes Project. I am Judy O'Rourke of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. We'd also like to acknowledge the C&EN Media Group for help with programming and promotion of the event. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Tim Hubbard. Dr. Hubbard is a professor of bioinformatics, head of the Department of Medical and Molecular Genetics at King's College London, and director of bioinformatics for King's Health Partners, King's College London. He is also head of genome analysis at Genomics England, the company set up by the UK Department of Health to execute the 100,000 Genomes Project, which aims to mainstream the use of whole genome sequence analysis for treatment in the UK National Health Service, NHS. If you would like more information about our speaker, please click on his photo image in the top right. The planners for this event have stated no relevant financial relationships. However, Dr. Kevin Davies did disclose that he is a scientific advisory board member for Pathway Genomics and is on the Speakers Bureau for Harry Walker Incorporated, and that neither constitute a conflict of interest. Although Dr. Hubbard has indicated that he has a relative financial relationship with Genomics England Limited, this does not constitute a conflict of interest because Genomics England Limited is wholly owned by the UK government and does not meet the definition of a commercial interest. I will now turn it over to Dr. Hubbard for his presentation.
Thanks very much. So I'm very happy to be here, sitting in Genomics England right now in London. I'm going to talk to you about the 100,000 Genomes Project. So firstly, I think it's useful to say, since this is an international audience, a little bit about UK genomics and healthcare kind of 101. So obviously, UK has been a leading figure in the Human Genome Project, with a third of the human genome being sequenced at the Wellcome Trust Sanger Institute, where I was head of informatics for many years. It's also, of course, led on the 1,000 Genomes Project, which was the sort of last set of large-scale genomes publicly available. Most recently, it's carried out the UK 10K project, which is a cohort of patients, cohort of individuals who have conditions where there is phenotype information. All of those projects, though, have been low coverage, at least the 1,000 and 10,000 project. The UK also has something called UK Biobank, which sounds quite analogous to the Precision Medicine Initiative 1 million cohort that's being planned in the US. So this is 500,000 individuals, 40 to 69-year-olds, and they were recruited, the recruitment finished in 2011. So those patients, they're not patients, those participants have been quite heavily phenotyped. They haven't been genome sequenced, although they have been genotyped. That's a collection which actually is available for research inquiries to carry out. So there's a biobank that we have in the UK. Now the National Health Service, the NHS. So we're a single-payer organisation in the UK. Obviously, everything paid through taxation. There's actually 4 services across the UK. So I'm talking about Genomics England, which is doing the 100,000 genomes in the zone of England, so under NHS England. And the health system is 110 billion annual budget, about 1.4 million employees. So it's a very large entity, the NHS, in England. So the history of this, there's a long history towards trying to link health and research and to bring in genomics. So many countries are interested in, can we use the information that's stored in electronic health records and get that available for research? And so there's multiple initiatives, but one of the most recent ones is the OSCE eHealth Board, which was set up to enable research on top of the investments in the UK in electronic health records. That led to the launch of the CPRD, the Clinical Practice Research Data Link, in 2011, and the founding of the FAR Institute, which is set up to build capacity around use of patient records. On the other side, there's trying to get genomics into health. So in 2009, there was a House of Lords report on genomic medicine, basically flagging that after the cost of the initial genome, now prices had come down to the point where it was feasible to start thinking about putting whole genome sequencing into the health service. That led to the creation of the Human Genome Strategy Group, which was in the Department of Health, to plan how you might execute genomics in the health service. A year later, the UK, the government changed and launched a life science strategy, which that was incorporating many aspects of trying to make the UK a better place for research in pharmaceutical areas. And a year later, an update of that, the announcement included the announcement of the 100,000 Genomes Project. So it was the end of 2012. 2013, Genomics England was launched as the entity to run the 100,000 Genomes Project. So the 100,000 Genomes Project, firstly, if you compare it with UK Biobank, 100K is a treatment project. It's really an NHS transformation project to try and get whole genomics into the health service. The project is entirely whole genome sequencing at a clinical grade, that's more than 30x coverage, focused on rare diseases in the form of trios, cancer in the form of normal tumor pairs. You can see the timeline. I've already mentioned the setting up phase. We had a pilot in 2014, but the main thing I'm going to talk about is the structure of the main program, which has been started since the beginning of last year. So it's got a number of objectives. Obviously, it's to sequence 100,000 genomes. But each of those genomes, it's hoped that they will lead to improvements of health for those individuals being sequenced. On the other side, it's also hoped that the existence of 100,000 genomes and the need to interpret that will stimulate the economy, stimulate the setting up of enterprises that will build experience in terms of interpretation in the UK. It's also meant to leave a legacy of infrastructure able to handle the huge amounts of data involved and human capacity and capability within the health service to handle that as well. And then finally, of course, it's a large data set. It's hoped that this will stimulate genomics research. Now, if you can think about the process for doing this whole business, starting from a DNA sample, ending up with clinical action. And you could look at that pipeline and build all the components of the pipeline. But Genomics England was set up as a lightweight organization, and the idea was that rather than build everything from scratch, it would outsource, procure components. And so to make the model rather like this, where you separate out the sequencing component from the interpretation component, and then the feeding back into the health service. So that's really what's been built over the last couple of years. You have Genomics England in the middle. First stage was, of course, to work out who was going to do sequencing. So we had a bake-off where we sent some samples to a number of companies. They returned sequence data on disks, and we assessed that for quality and coverage. And that informed the contract, which was eventually awarded to Illumina. So one of the principles behind this whole project is that the samples, the data doesn't move out of England, that it's a health service project, so in terms of privacy, everything must happen within England. And so this involves Illumina making investments into the UK to build a sequencing facility that's actually going to be hosted at Hinxton, where the Sanger Institute is, in a building that's being built by the Wellcome Trust, that is being leased to Illumina to fulfill the contract. So the next thing you need, which is arguably harder, is to get a flow of patients, consented individuals, where their sequencing by whole genomes will lead to an improvement in their healthcare. And so this is being managed through, in the main project, through NHS England. So NHS England has contracted the creation of a number of genome medicine centres in the National Health Service. So because it's a National Health Service, the aim is for to have access to this across the country. And so these are essentially regional centres that have been selected. There's a lead organisation, but then there's local delivery partners. Involves around 90 hospitals. It's about a third of the health service now involved at some level in providing the flow of consented individuals into the programme. So then the other thing you need, of course, in this particular area is somewhere to put the data and handle the data. And so, again, this is procured through, in fact, a government framework, because the government has its own secure data centres, but these are now structured around private cloud computing. So commercial providers will provide the infrastructure under contract within a secure centre. So that's the model that's been adopted. Now, so you have now the flow of DNA to Illumina, and you have the flow of data into the data center. There's one other thing that's being taken out from the Genome Medicine Centres, and that's phenotype data. And this is a major other side of the program. We don't just want to collect samples, we want to have a very clear statement of the patients who are being brought into the program based on unmet clinical need. So the idea is there are already existing processes in the health service for doing single gene tests, panel tests, and we want patients to have already gone through those processes so we don't end up sequencing whole genomes when there's an existing test which can be deployed. So there's an eligibility criteria attached to all the rare disease conditions, for example, that we are currently allowing to be brought into the program. Patients who meet that eligibility criteria, there also needs to be data collected to make it very clear what the phenotype is of their condition. And that's linked, each of those disease areas also has a gene panel associated with it, which is also curated. So the data models, they're really to capture phenotype information. So HPO is one standard part of that. Definition, each of the data models has a clear definition of what phenotype terms we'd expect. And the way this is all managed is through a data model catalog, which holds all that data in a database and can be curated so that we can evolve these models over time as it becomes clearer from a combination of views from experts, clinicians, and researchers. What's the best set of data to allow each genome to be interpreted and also being used for research. Now, the way that database operates is that the curation can be exported in the form of XML specification of the data that's required to be reported, and that can also feed into a clinical interface. So the Genome Medicine Centres, they can populate this data for each of the patients they've consented, either through this clinical interface based on Open Clinica, or they can write an XML specification directly out of their electronic health record system. So this, we're basically supporting extraction of data from electronic health systems into this program. So that gives you the feed of clinical data. Now, we also are inputting various other types of data which is collected elsewhere in the health system. There's hospital episode statistics. There's a registry which collects, for example, cancer registry information. That will also be brought in, but as a sort of secondary research data source. The primary data that we need to interpret those genomes is being collected directly as I've described. Now, once you've got all that data in the data center, obviously you need to interpret those genomes. And again, rather than building our own interpretation pipeline, we're managing a series of interpretation companies. We've been doing that through another bake-off, another competition to evaluate different groups in this field. So back in 2014, we ran a competition with 30 different groups taking part. We made a short list of 10 groups, and we've now selected from those, offered initial contracts to provide those interpretation services. Now, I mentioned earlier on that there's this intention to stimulate the economy. So there's another parallel effort involved of providing grants to a few companies to stimulate activity in the UK in development of this sphere of genome interpretation. So those companies that have been selected, they run their interpretation services within the data center. And it's the same model, just as the samples can't go out of England, Illumina has to be based in England. Here, the data doesn't go out of the data center. Any service has to run inside that data center to provide a clinical report. Now, the feedback to the NHS, it's not analysing a whole genome for all the possible future conditions. It's very specific. These are all ill people with clear phenotypes, and we want primary findings to diagnose their actual condition. There's a limited number of secondary findings which can be reported, but that's based on the consent of the individual. That's an option for the individuals to choose whether they want these secondary findings. But it's a very, very limited set where there's actionability in the health service, and it's a very clear risk allele. Now, some figures. So we're currently up, this is a bit out of date, it's more like 6,000 whole genomes that have been sequenced and transferred into our data center, and there's more, obviously, patients that have been consented through the initial 11 genome medicine centers. The genome interpretation providers, 3 out of 4 of these 4 that have been selected have been brought into the data center, and one of them is actively running. Genomics England is providing the wrapping around that. The companies have to produce reports in a fairly standardized way, and they also have to combine that with information from gene panels, genes that have been curated as being likely to be associated with each condition. So this produces a degree of standardization despite the fact you have different providers. So essentially we're going to provide reports back, but also do an evaluation in this first 8,000 genomes of how these different companies perform. And we'll be looking for new companies in the future, bringing in additional testing of other providers over time. Now, I mentioned the model development. I mentioned the model catalog and how that's curating the gene models. Similarly, the gene panels are being curated, the sets of genes that are thought to be associated with each disease. And so there's a panel app which you can view, and you can also apply to be an expert reviewer. And this is basically has all the diseases that are currently in the eligible data set for collection, and it has ranking of how people have reviewed the different genes that are associated with each of those conditions. Now, this brings me to, because all of those experts, they're a mixture of clinicians and researchers, and that links to the research part of this whole design. So an important part of the 100,000 genomes is that it's all under a research protocol under a new designation, bioresource in the UK, which allows us both to do clinical treatment and to do research. And so that allows recontacting, collection of an extra biobank sample, which is available for future omics studies. So it's a real combination, one data set being used for two purposes. And if you look at this diagram, so this now extends this diagram, so the way that the research is being organized is through what we're calling Genomics England Clinical Interpretation Partnership. So this is a whole load of PIs, about 2,500 PIs, mainly from the UK, but also outside the UK, which are coming together to form domains working in particular areas, which I'll describe in a minute. The main principle is that those partnerships, they also have exactly the same constraints. The analysis has to be done inside the data center. There isn't any export of that data. There's a similarly, and that data, that consortium can also access the biobank repository sample as well. They can propose investigations that they might do using that sample. Parallel, there's also something called the Gene Consortium, which is a set of companies, and they're running exactly the same way in a pre-competitive framework, accessing data, but again, they can only analyze data from within the data center. So the clinical interpretation partnerships divided up into domains. So there's a set of specific domains around particular diseases, so in cancer and rare diseases, and then there's cross-cutting domains looking at different generic analytical processes using the data to try and improve those processes. The goals here are obviously doing basic research, but they're also to drive up interpretation of those individual genomes. So here we have, we anticipate maybe the diagnostic rate will be increased from what it is right now, which is probably around 20–25% of individuals using the existing diagnostic tests in the health service to maybe 50%. But that still leaves 50% of individuals who aren't going to get a diagnosis because it's not clear looking at their genomes where the actual variant of interest is. It's hoped then that the clinical interpretation partnerships will be able to look at those groups of variants across groups of individuals and improve the interpretation, improve the curation of which genes are involved in those conditions. So up to now, we've launched a set of domains, we've approved a set of domains around the different cancers, the different rare disease areas, a number of cross-cutting domains, and an ethics domain and a validation domain. And this will be evolved over time. At the moment, these groups are just getting going in terms of working out their research plans, applying for external funding, etc. Now, so if you look at this whole model, I think you can see that you can kind of simplify this down. Basic premise behind this is one set of data on the health service side where it's used both for interpretation and research. So that's the kind of model we're building here, and it can be potentially a future way of doing health service research and treatment in a combined way. Now, it does lead to considerations about data sharing. For example, we've been used to, in the Human Genome Project and the 1,000 Genomes Project, being able to just download data and run analysis whenever we like as researchers. It's got slightly more difficult with these large-scale data sets, which include phenotype data, such as any of the studies which involves patients with phenotype information, because they're stored in dbGaP or EGA, and you have to go through approval processes to get access to that data. You have to show that you can hold it on a secure system, for example. In this case, in some ways it's harder, in some ways it's easier, because you won't be able to download the data. And that, in some ways, that's more practical, because it's easier now to bring the algorithms to the data because the data is so large. But it also means that we're protecting individuals' privacy by telling them that the data isn't going to be distributed, so they have confidence that we know what the data is being used for, that we can watch researchers. Researchers can also work in a unified environment and deploy their algorithms there to do analysis. So that's the model that we're moving towards. We think it's going to be a future model for dealing with healthcare data. Now, one of the risks it is that it will generate a set of walled gardens, and of course, in very rare conditions, it's pretty valuable to be able to collect data from around the world, as projects like Decipher have managed very successfully to do. But it should still be possible to bridge that gap, even if you can't distribute the raw data through federated analysis. And so there is now this organization called the Global Alliance for Genomes and Health, which is building software infrastructure, has already deployed things called beacons, which make it possible to have closed data sets which can be asked questions which don't reveal information about the individuals in those collections, but do allow a degree of data or information sharing. So I think that's the kind of evolution of that pipeline, is the sort of future to making sure we can do effective information sharing without sharing the underlying raw information that protects people's privacy. Now, there's one other aspect I haven't mentioned, which is worth pointing out. I said that the health service in England has 1.4 million people. If the 100,000 is really just the vanguard of moving into whole genome sequences in a health service, then we have to do training. And so part of the money that's been allocated, £100 million that was initially allocated to Genomics England, went to Health Education England to provide MSc courses for teaching. And this has also been deployed across the country. You've got 9 different universities, including my own King's, teaching MScs specifically around genomic medicine. So the overall picture is that this project, 100,000 project and Genomics England, it's providing a transformational catalyst for the NHS through the Genome Medicine Centre contracts. It's leading to standardization of secondary care data collection within the health service, and piloting this dual-purpose data center model to allow secure usage of clinical information and provide a way of deploying interpretation services, both for research and also for treatment. Now, if I put my other hat on, because I'm at Genomics England part-time, I'm also at King's Health Partners and King's College. If you look at it from the point of view of the Genome Medicine Centre there, well, there's many advantages of being part of the 100,000 project and the sort of infrastructure that's being constructed for the future. So this brings us in to the expertise of using very large, whole-scale, whole genome data in a health system, because King's Health Partners, of course, is one of these academic health science centres that is a university and a set of hospitals. It takes it into these national data sets, as opposed to just our local data sets, for analysis. It does drive the leveraging of electronic health record systems, to be able to support the requirements of the contract for Genomics England and NHS England. It's leading us to think about better ways of structuring our EHR so that we can capture data in a more systematic way as part of the drive in the UK as a whole. But I think finally one of the interesting things is it will drive the possibility of developing your own apps for decision support, because now you're going to have a set of genomes stored, which are potentially available on demand for interpretation. It's a small set right now because it's just the 100,000 data, but data is going to be stored and is potentially there so that it can be reused if a patient comes back with a different condition, or even something where there's a potential for pharmacogenomic analysis of which is the most appropriate drug. So I think if you take this model, you can think of adding one additional line, which is where the Genome Medicine Centres potentially fire off apps which run in the data center on the genome and provide a decision support output that feeds directly back into the EHR of those medicine centers. And in terms of the way of thinking about this, if you think of research that tends to happen in hospitals and university settings, it's about collections of samples. It has been about collections of samples where you've got partial data, genotypes, exomes, low coverage genomes, and doing analysis across those to find patterns to look for disease relationships. But we're now going to switch to this, where every sample is sequenced at a clinical grade. And so you can still do that analysis to look for new disease relationships, but you can also look at every individual and say, what can I say about that individual that can feed back into their clinical treatment as well? And I think that's another direction of research that we should expand to encourage people to think about interpreting individuals and feeding back directly into their treatment, as opposed to just looking for patterns which will feed a drug development pathway in a very slow, maybe 20 years ahead sort of research purposes. So that's a 100,000 genomes project. 100,000 whole genome sequences being collected in a healthcare system. It's working as a collaboration across the NHS, industry, to embed genome medicine into the NHS. It includes educational support, and it's meant to leave this legacy structure in the health service. Genome Medicine Centres, computational infrastructure, and a way of reusing genomics data. And it's hoped, of course, it will lead to new diagnostics and therapies and new opportunities for patients as part of essentially personalized medicine. Everything initially has to be done by the end of 2017. That's the original structure. But actually, Genomics England is now extended, at least until the end of the next 5-year budget cycle, to the end of 2020. So, in fact, this is really the beginnings of a national infrastructure to support precision medicine, genomic medicine in the UK. So there's lots of acknowledgements. Lots of different groups across the UK have contributed to the initial pilot, which I haven't really talked about, but it's the data that's been collected in the first few thousand genomes. There's now quite a big team here at Genomics England of about 100, more than 100 people, and there's a large number of external experts who are involved, almost continuously in advisory committees and working groups. So it's a big national enterprise now, which is beginning to produce results. We have fed back some initial results, and we'll start the pipeline of feeding back individual interpretations to patients over the coming months. And I will stop there. Thanks very much.
Thank you for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, short reads miss a lot of information. Do you think the UK has to sequence those 10,000 again using Nanopore or PacBio long reads in the future?
I'm back on the air, am I? Right, so the short reads, obviously it's not perfect, but it does produce certainly a high quality. I mean, part of the criteria for sequencing is a quality threshold in terms of coverage. We're obviously going to have problems in areas of structural variation, to a lesser extent in CNVs, but certainly most of the variation we're going to capture. Are we going to have to do this again? Remember, this project as it's set up right now is to provide treatments to those individuals. So if we get a diagnosis, then that's fine. The additional benefit of looking at those genomes, they'll be stored. Maybe in future they'll be resequenced. But the primary drive right now is to use the sequencing technology that's available on the basis that it's good enough to have value right now.
How are patients involved and empowered? How are they educated? Can they communicate and collaborate with others in their phenotype, genotype to compare notes, to add more lifestyle context, etc.?
There's a substantial activity to engage with patients, and in terms of having an interface, of course, this is all through the healthcare system. So it's through the clinicians in the Genome Medicine Centres. Now, there is a national program under the health service to, in the next year, provide everybody with access to their patient records. and to leverage some support on top of that. So in some ways, a lot of this is going to happen at the health service side as opposed to purely Genomics England. But we are in discussions around enabling patients to contribute information as well. But right now, you'll remember, if you think of 23andMe, most of those people, they're not ill, they're interested in their health and they're interested in their genome. Whereas here we're talking about health service patients under clinical care, and literally a lot of them, they just want a diagnosis.
How many of the 5,000 whole genomes sequenced came from healthy volunteers? And in addition, would you encourage blind screening of the general population in England above a certain age?
So as I said at the beginning, we have a cohort of healthy volunteers. That's the UK Biobank, and it's not sequenced yet, but it is. It's a comparative cohort, some of which will get sequenced, of course. So that's, as a population, here we're not sequencing any controls except the parents. When we do trios, we're basically only dealing with people who are ill in the healthcare system.
Is there any scope for other private genome centers in the UK to play a role in this project in terms of sequencing?
Right, so in terms of private genome centers, basically, no, this is really being done as a centralized structure. Now, of course, we're at the stage right now where we're only doing 100,000. I think as the health service, assuming the health economics on this all looks good in the future, then I think we will be, we will end up with more than one sequence provider. But right now, it looks that the most cost-effective way is to have a degree of centralization, even though you have to transfer the samples. You have to build this infrastructure within a health service to manage this. But this seems to be the best way of doing it right now. Okay, I can look at these things now, so I'll answer the next question. Is it feasible to go? Is it feasible to go for 30x coverage for cancer? So I must admit I didn't point out that for cancer we're sequencing much more than 30x. For the tumor sample, it's more like 75x. The 30x is for the rare disease studies, and that's based on the idea that when you do 30x as an average, you can set a very high level for a very high fraction of the genome, more than 95% at more than 15x. 15x is what you need to confidently call hets in a large number of cases. So that's the kind of criteria wanted. Right, the computational environment. We are actually investigating using Hadoop internally because if you think about 2,500 PIs coming in with their postdocs and students wanting to analyze this data, we're trying to provide interfaces within the system to support analysis. One part of that is to read all the variants into a database based on Hadoop, so it can be rapidly queried, so you can query subsets. So that's a particular project being led by the Director of Bioinformatics, Augusto Rendon. In terms of the general computational, everything is really worked around virtualization, virtual machines, a Docker environment, so that everything that people bring in has to be packaged so that it will run inside the data center with minimal intervention. So I'm being asked about what kind of international collaboration is happening in parallel. So there are a couple, I've mentioned already, the Global Alliance for Genomes and Health. That's really a project more around how can you share data. And Genomics England is part of that. There's quite a lot of UK institutions, including King's College. Specifically for projects like the 100,000 genomes, which is essentially a treatment project, a genome medicine project, there is another organization called G2MC. So that's the Global Genomic Medicine Collaborative. So that's a set of national organizations that have met a couple of times. Last time was Singapore in November, looking at how genome medicine techniques are being deployed in healthcare systems. And there are essentially the UK is almost unique in doing whole genome sequencing directly in a healthcare setting, but there are other countries that are deploying specific, whole national screening processes using genomics, for example, very specifically around detecting Stevens-Johnson syndrome, which is extremely severe immunological reaction to certain drugs that leads to essentially a very high death rate as an immune reaction to the skin, and you can screen for that. And in terms of the health economics, it looks like in some countries where it's highly prevalent, in Asia, it's worth screening the population in order to detect that. So those kind of projects, are looking. We're part of that sort of grouping to exchange expertise around how do you deploy this stuff in practical, including children. So again, I emphasize that the structure here is around unmet clinical need. So the types of patients that you would see normally in a clinical genetics units of a health service, they're the ones that have this current diagnosis rate through single gene tests or panels, and it's those that are eligible if there's a clear phenotype and it looks like you have a good chance of providing an interpretation using whole genome sequencing. And so that includes children where it's relevant under those conditions. In terms of cancer and epigenetics. So the point here is, so firstly, cancer is coming on much more slowly than rare disease. The project is really focusing on rare diseases at the moment, but that's partly around the difficulty of, we're talking here about standardizing clinical processes in hospitals. It's been quite challenging to standardize in rare diseases. It's much more challenging when you deal with the variation in pathology services and the problems of the current processes that are used in cancer with formalin fixation, which makes it difficult to sequence. So those are the problems that are affecting the cancer program right now. Now, do you need to do RNA? Do you need epigenetics information? This project is very specifically only to collect whole genome sequence, but we're biobanking a sample that's going to be available for follow-on investigations on the research side. Now, we also are investigating doing RNA in parallel with whole genome, particularly for the cancer interpretation. I think, so this is a question now about, again, cancer against engagements of the patients. I think, this is basically up to patients what they want to do. There are a lot of groups that exist already to share information between people who want to exchange information about diseases. And, there's nothing to stop them doing that. I'm just saying that the health service as a whole is starting to share more information, but it's not specifically set up to build a social network. I think others will end up doing that.
I would like to once again thank Dr. Hubbard for his presentation. Do you have any final comments to share with us today?
That's fine. I think that's just a. I think just to say that it's a very exciting project, and I think if you look around what's happening in England, in London, we have things like the new Crick Institute just starting. In fact, some part of London is now referred to as Tech City because it has this huge collection of technology startups, around 2,000 in the sort of area, actually roughly around Genomics England. We have another side around the Crick Institute, which we're now referring as Med City. So this business of stimulating SMEs around doing things with health service data to improve healthcare delivery and to personalize it. Genomics is one component of that, but actually England, the UK, London is really a great place to do that kind of activity now. So anyone who's interested in being, pushing this frontier, and it really is a frontier that we're pushing, come and look around and see what it's like to come and work here. Thanks very much.
Thank you. Today's webcast will be available for on-demand viewing through August of 2016. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that email, that announcement, to your colleagues who may have missed today's live event. See you next time. Goodbye.